All right, so in case you're wondering, my daughter survived skydiving. Probably figured that out. I wouldn't have been here on Tuesday, but she didn't. So just to let you know, all is well there. And thanks for some of you that encouraged me that she would probably be just fine. Also, little public service announcement. I had a few people mention to me on Tuesday that they were really uncomfortable with certain students not wearing their masks over their face. So I know that you have to kind of bunch up before an exam. I think for the next exam, I'll just let everybody in and I'll hand it out one by one. We'll have plenty of time, I think, to do that. And I know you have to bunch up a little bit when you leave the class. Of course, we had a lot more people here for the exam, but just out of, if nothing else, out of respect for other people feeling comfortable, if you could just make sure your mask is on all the way. Cases in Michigan are rising, so you know, I don't know what's going to happen with that, obviously, but just wanted to mention that. I know that it's been a tough week for all of you, so just try to encourage you to hang in there. It's a busy time, a lot of exams. We have about 5 weeks to go till Thanksgiving, so it's sort of on the horizon. And of course, after Thanksgiving, I'm not sure if it's a university-wide policy, but for this class, we will be virtual after Thanksgiving. So how that will look, I'm going to be recording lectures for you to view. And during class time, I'll have open Zoom meetings if you want to ask questions, you know, basically kind of like office hours, but you can plan on that and, you know, ask me whatever. We can go over things again, anything you want. But and then the final will be by respondus. So plan ahead for that if you need to, you know, figure out a computer or whatever to do that on, but just kind of looking ahead there. So I had a few of you that bubbled in your username incorrectly or not at all, and I think I have figured out who those people are. I had a few people that it seems like didn't show up for the exam, so if your score has not shown up on Blackboard, get in contact with me. I have a couple people yet to take it, but this is what I can tell you so far, the distribution of the grades. It was about 72 before the curve, and I just gave a 1% curve. I know that's not a lot, just to bump up a little bit. It's about, you know, high 73 after you know, the rounding and all that. So actually, this was really pretty solid exam. You know, I've had to curve a lot more than that. So overall, pretty happy with it overall. Do talk to me. I've had a number of you talk to me, you know, strategies to do better. By all means, come in and look over your exam. I had 3 or 4 people that only missed one. No perfects this time. So again, pretty good overall. Okay, so we will forge ahead. We are really kind of switching gears here a little bit. Up till now, we've learned all about the structures of microbes and all different kinds of microbes, and how they get energy and how they reproduce, how they grow. We're really switching gears now to thinking about how to kill them. And I don't want to come across as all these microbes are bad. I know that's kind of the way we focus, all right? They're necessary for life, and we'll talk more about how our microbiome is really necessary for our health. But we're going to concentrate for a while on how to kill them. Chapter 9 is how to kill them in our environment. Chapter 10 is how do we kill them with antibiotics when they're in our body. And then we think more about that process of infection and how our body's immune system works to kill invading microbes. So really that's what we're focusing on for the rest of the semester, and kind of drawing in those things you've learned about the structure, mostly about bacteria, but viruses and other things as well. All right, so if you think about historically how people try to keep themselves from getting sick, They had no idea what was making them sick. The kind of biggest theory for centuries was bad air, miasmas. People seem to get more sick when they were around a swamp. It's because there's lots of mosquitoes around swamps, and mosquitoes carry lots of disease. But they had all sorts of, superstitions about how to keep away disease. Bathing in urine was one. I read about tying live chickens, a live plucked chicken to you to try to keep, ward off the plague. Toads around your neck. Of course, we didn't know how to treat it when somebody was sick either. So for a long time, they were using leeches to suck out, the blood that they thought was bad. And personal protective equipment during the Middle Ages or 14th century looked something like this. All right, they had this beak that doctors put over their mouth. They put herbs and things so they couldn't try to keep away that bad air. I don't know, maybe that included, maybe protected them a little bit, but certainly looked imposing. All right, so chapter 9, here's some of the topics we're going to cover here today and into next lecture a little bit. So we're going to talk about the differences in susceptibility of organisms to antimicrobial agents. That's kind of a general ladder of things. Yeah, thanks for closing that door back there. We're going to talk about some terminology and things that we have to be straight on first. Some physical means to kill microbes. Heat and radiation are two big ones. We're going to mention some other ways as well. We're going to talk about lots of chemicals that we use and how appropriate is that today in the midst of a pandemic. We're trying to decide the best way to kind of keep ourselves safe. Hopefully you'll pick up some tidbits that you didn't know before about chemicals that you've used before, like bleach. Why do we keep that in a white container? And near the end of our discussion here, we'll talk about the best way to wash your hands. So maybe you've read something about that already, but interesting to look at. All right, so I like to start out with just some terminology. And your book has a great table in it that goes over some of these terms. So I'll just kind of go through this. And I know you can read it yourself, but I'll kind of highlight a few things here. Some of these terms you've probably used for a long time and maybe haven't used them quite in the correct sense. So we will try to use them correctly. These two terms, and these are in alphabetical order, which is not maybe the way that I would put it, but if you, the difference between antisepsis and disinfection, basically both those terms are referring to reducing microbial numbers. The difference is where you're reducing them. So an antiseptic measure means that you're reducing microbes on living tissue. So an antiseptic is designed to be used on living tissue, something like soap, or maybe iodine before a needle poke. Mouthwash is considered antiseptic. And then a disinfectant is designed to be used on non-living tissue. So what you spray on the benches in lab, what you put, in your pool water to disinfect it, for example. And typically disinfectants are going to be a little bit stronger because, again, they don't have to be designed to be safe to be used on our skin. Aseptic, if you are in lab, that's a term that hopefully you recognize. We try to follow aseptic technique in lab. That just refers to a technique where we're trying to reduce any contamination. So again, kind of cut down on microbes that we don't want growing. So aseptic technique involves things like flaming your loop before you use it to inoculate a culture, or wiping down the bench with bleach or alcohol, keeping the lids on cultures when you're not using them. These suffixes, side and sidal, if you compare those to stasis and static, if something is a bactericide, that means it's designed to kill bacteria, or a fungicide designed to kill fungus. If something's bacteriostatic or a bacteriostasis treatment, that means it's just going to keep the bacteria from growing. So a good example of a static measure is refrigeration and freezing. Okay, you put those organisms in the cold and that doesn't kill them, it just keeps them from growing. Okay, where a lot of chemicals are designed to kill that organism in question. This term degerming mostly is referring to a mechanical scrubbing, trying to get rid of microbes that way. So it usually, involves soap or something, but it's kind of all about the scrubbing. And then pasteurization, we've talked about that. We're going to talk about that in more depth here in a little bit when we talk about using heat. But that's just a treatment to reduce microbial numbers. Louis Pasteur first did that to try to control the spoilage of wine in France. Pasteurization is a sanitation. It's designed to reduce microbial numbers. And usually we use this word sanitation when we're referring to some public health standards. So restaurants have to have a little higher standard of cleanliness. If you've ever washed dishes in a restaurant, you have to wash them and rinse them, and then you have to soak them in a bleach solution for a while. So you have this kind of extra step of sanitation. Okay, and then sterilization is some treatment that destroys all microorganisms, so kills everything, including spores. Typically, to sterilize something, most often we're going to use heat. We do have a few chemicals that act as sterilants, but they're kind of only a few. We call them high-level chemical antimicrobials. So, all right, so a little background behind some of those terms, again, that we'll try to use correctly. So we always try to think about how these agents are actually killing organisms. Sometimes we don't know exactly, but oftentimes we kind of have an idea of what's going on there. So there's two basic methods or mechanisms behind how these antimicrobials actually kill cells. One is that they alter cell membranes and cell walls. Okay, and that might not seem like it's enough to kill a cell, but think about what we've learned already. If that cell wall has some kind of holes poked in it, then it's going to be really susceptible to an inrushing of water due to turgor pressure. So most often cells are going to be in a hypotonic solution, and if their cell wall is, again, not firm, then they're going to be susceptible to water rushing in and the cell bursting. If the membrane is not intact, then things can enter or leave the cell that wouldn't normally do that. We just got done talking about the proton motive force and how that's so essential for the cell that's respiring to be able to make ATP, because you only want those protons flowing in at that enzyme, the ATP synthase. If it's flowing in other places, then it's not going to be able to make the ATP. And of course, in the case of viruses, if they're enveloped and that membrane gets broken down, then they're not able to attach to certain host cell receptors. So they're not going to be able to attach and infect the host. Okay, the other mechanism is that the antimicrobial agent damages proteins and nucleic acids. Okay, so we know that proteins, it's crucial that they're folded in just the right way. If certain bonds are broken, they're gonna misfold and not be functional. There are some antimicrobial agents that particularly are gonna target nucleic acids and cause certain kinds of mutations. So, all right, so the whole point of this discussion is trying to help us determine what's the best way to cut down on microbial growth. And there's not going to be usually any one best way, but there might be a way that's kind of best way for everything. But looking at certain circumstances, we're going to try to take a bunch of things into account to help us make good decisions. Okay, no one size fits all. So things we have to think about is the site to be treated. So for example, is the surface that you're trying to reduce microbial growth on, is it porous? Do you care if it's stained? You know, in labs, should we be using iodine maybe instead of alcohol? Well, iodine stains everything it touches, so no, probably not. Okay, so if you think about the surface, is it living? Is it non-living? That's a big one as well. Exposure time. Most of these treatments that we're going to talk about require some exposure time. Even antimicrobial chemicals that you would use in your home, if you read the label really closely, most of them say spray it on, leave it on for 30 seconds, and then wipe it off. A lot of us don't always follow those directions. So usually you need some kind of contact time there for it to work. You have to take into account the concentration. Ideally, if something's really dilute and it still works, that's better because it's going to be cheaper, but it doesn't necessarily follow that the more concentrated, the better that it will work. There's one exception there that we'll talk about. Are the cells that you're trying to kill actively growing? Okay. They are going to be easier to kill if they are actively growing. If they're in some dormant state or some biofilm, they're just not metabolizing very quickly, then they're just going to be harder to kill. All right, what's the temperature? According to this graph that's in your book, and I don't know what kind of cells these were used or where the cells were, but it's looking at the effect of some chemical on some cells and how fast the cells die at room temperature. It takes about 10 minutes to kill the cells. If it's 45 degrees Celsius, which I don't know what that is in the Fahrenheit range, but body temperature is about 35, 37, so it's going to be in the hundreds at least. The cells die a lot quicker. So their metabolism is quicker, more likely, and they're quicker to die. What about the pH? For some chemicals, that's going to be really important. If you've ever taken care of a swimming pool, then you know that's the case. You always have to test the pH because the chlorine that you add will dissociate, as the pH increases. And when the chlorine dissociates, it's not doing any good anymore to, kill, the algae or whatever it is you're trying to kill. So sometimes you have to pay attention to the pH. And a really important one that is often forgotten, is there organic matter present? Okay, so what I mean by organic matter, it could just be dirt. It could be things like blood or feces, diarrhea, bodily excretion. And anytime you have to clean up a mess like that, you can't just spray bleach on it and wipe it up. You usually have to clean it up first and then do an extra step of disinfection because a lot of our antimicrobial agents don't penetrate through organic matter. Okay, so I'll just write that in. Many antimicrobials don't penetrate. Okay, and that's something that's easy to forget. Hand sanitizer actually doesn't do a lot of good if your hands are dirty. Okay, so that's something to keep in mind. All right, also we want to keep in mind, what are we trying to kill? All right, so here's a very interesting chart from your book here. Keep in mind that this chart is kind of generally speaking, so there's going to be exceptions to the order of these things. I would say don't memorize this, just kind of have an idea of the kinds of organisms that are really hard to kill and the organisms that are really easy to kill. Yes?
Before you said that gram-positive bacteria were usually hardier than gram-negative.
So the question was, aren't gram-positive bacteria hardier than gram-negative? I'll get to that because that has, I've puzzled over that a little bit too. So hold that thought. Hopefully I'll come back to that. So yeah, keep in mind that this is generally speaking, and I believe that this is mostly referring to outside of the body. The difference here between, you know, gram-positive and gram-negative, gram-positives of course have the thick heteroglycan cell wall. I think this is, that they're saying gram-negatives are harder to kill because I think that they're talking mostly inside the body. Because again, that outer membrane, a little bit more impermeable to a lot of things. That's how I explain that. I'm not sure if there's really any difference outside in the environment in ways to kill them. So that's a good point. We've talked about how enveloped viruses are really wimpy in the environment. Now, I just saw something on the news a couple days ago. Some researchers had said, oh, coronaviruses are surviving for 25 days in the environment. And I thought, I don't believe it. I just don't believe it. It's an enveloped virus. I think it's a fairly small, well, it might be on the small side, but that shouldn't really matter whether it's a small envelope virus or a large envelope virus. I would like to see that reproduce several times before I believe that study. Because usually envelope viruses, it's a number, you know, hours, maybe a day or two on a surface in the environment. Again, because that phospholipid membrane just dries out really quickly in the environment. Really sensitive to, you know, sunlight, UV light, anything like that. And if a virus, again, loses its envelope, then it can't attach, it can't be infective. On the other end of the scale here, prions, we talked briefly about prions. Remember, they're just proteins. How do you deactivate a protein? Well, it takes heating it up to 900 degrees for 4 hours to inactivate a prion. So they're really hard to kill. We do have some chemicals that can excuse me, inactivate prions, but you have to, you know, soak the medical instrument or whatever in the disinfectant for hours and hours to clean them. Okay, luckily we don't normally need that level of disinfection. Prions are pretty rare. Of course, bacterial endospores are difficult to kill, more so than vegetative cells. We talked about that. It's kind of interesting to look at this chart because you should be thinking, wow, I know all about this stuff now. We've learned a lot so far. We talked briefly that some protozoa can form cyst stages. Cryptosporidium is not a protozoan that we talked about. That's a common, I don't know how common it is, but it's basically lives in fresh water, and it's impervious to chlorination. So that's why a lot of our treatment plants now, we don't use chlorine, we use ultraviolet light, and that cysts are a little bit more sensitive to ultraviolet light. But it's kind of interesting to read case studies sometimes. Back in the 90s, there was like tens of thousands of people that got Cryptosporidia. It's a nasty diarrhea. because there was a little glitch in the water treatment plant. I think it was in Milwaukee, Wisconsin. We gotta, that's something that, you know, we want to protect against. They're hard to kill. Of course, the mycobacterium, because they have the acid-fast cell wall, just makes them a little bit hardier in the environment. And again, in here, there's going to be a lot of variation depending on certain organisms. So try to just, you know, the hardest to kill and the easiest to kill are mostly what we're kind of thinking through. All right, so the actual methods that we're going to talk about, when I listed them, you know, neatly on one side of the other in your notes, when I tried to do that here, it kind of goofed it all up. So I had to write it in a little different here. But we're going to start out with some physical methods of control, heat, some other methods we're just going to mention kind of briefly, and we're going to spend the most time on heat and radiation. Okay, those two things there. And then we'll start thinking about some different chemicals. And I have those arranged, I think it's a little bit different than the way the book arranges it, but low, intermediate, and high level. Basically, a high level antimicrobial chemical is going to be one that's sporicidal, okay, that can kill endospores. And that's the big difference there. But the low level ones kind of work on the lower level organisms here on the chart. And as, you know, they get intermediate and high, they're going to work on organisms a little bit higher up. Okay, so that's sort of where we're going, the actual methods that you will, I guess, be responsible for. Let's start out by talking about heat. That's usually the first thing that you think about, kind of really the best way to cut down on organisms' growth. Of course, we're talking about on inanimate objects. We can't very easily use heat to kill something, you know, on our skin, with the exception maybe of burning off warts. Of course, warts are caused by viruses, but other than that, I can't think of really any applications there. Okay, so why does heat kill microbes? Basically, does a little bit of everything. So denatures proteins, destroys membranes, cell walls, disrupts the structure of nucleic acids, pretty much just everything. Okay, and we do have a couple of ways that we can measure an organism's tolerance to heat. Okay, the first way we call the thermal death time. And this is something in a regular semester you would determine this in lab. We have a whole kind of heat tolerance lab. So I decided to talk about it in a little bit more depth here. The thermal death time, we define that as the shortest time that kills an organism at a given temperature. Okay, so you're keeping the temperature constant here. So let's say 65 degrees Celsius. That's pretty warm. Remember, boiling is 100, so it's not that warm, but much higher than body temperature. And you would put, say, 3 organisms that you're testing in a broth test tube. You take out a time 0, just make sure that they're viable. Maybe put a little bit on a plate and incubate it, just see that it's growing. You expose that test tube for a couple minutes, probably to a hot water bath, set at 65. After 2 minutes, take out an aliquot, put it on a plate so you can see whether it was viable, you know, and then test it for lots of different time periods. Again, taking out an aliquot, exposing it to that 65. So you let that plate grow up overnight that you were taking the aliquots out and see if there's any growth there. All right, we do see a difference in organism susceptibility to heat. In this case, we always expect bacillus to be really tolerant to the heat because bacillus is a spore former. So it's not going to form spores in 4 minutes or 6 minutes, but if there were spores already in that medium, they would survive that heat exposure and then germinate when we, you know, incubate it in a temperature that it likes a little bit better. Okay, so in this case, if we were to determine the thermal death time of these 3 organisms, for E. coli, it would be 4 minutes. For Staph aureus, it would be 4 minutes. And for Bacillus, the best we could say was greater than 10 minutes. Okay, we would have to test it a whole lot longer. Okay, the other way that we often represent an organism's tolerance to heat is by the thermal death point. So with this measure, we're just switching the variables here. We're keeping the time constant. Usually we would say 10 minutes as the time, and then we're varying the temperature. So the lowest temperature that kills at a given time. All right, so in this example, again, we've got the same 3 organisms. The thermal death point for E. coli would be 60. The lowest temperature that it kills for Staph aureus, 65. And again, Bacillus is surviving a lot longer because it's a spore former. So the best we could say is greater than 70 degrees Celsius. Okay, now spores are not impervious to heat, but they do survive a lot longer than vegetative cells. Okay. All right, so some different applications of using heat. Okay, the first one that everybody thinks of is boiling. Okay, that's an easy way to disinfect objects at home, you know, baby pacifiers or canning jars or whatever. It's actually considered a disinfection or a sanitation. Again, those 2 terms are really similar. Sanitation is usually referring to some public health, public setting like a restaurant or a hospital. Okay, it's considered only a disinfection because it actually takes longer than 10 minutes to kill spores. And I've heard different numbers as to how long it takes to kill spores, you know, half an hour, sometimes even longer than that. So actually that's kind of surprising to me, but that's why it's not, boiling is not really considered a sterilization. It's kind of the best we can do, like at home usually, kind of the best method that we have. And the problem is that things that you boil often become contaminated really quickly again because they're wet. So again, but it's going to cut down on most organisms, anything that would probably cause an infection. Another application of using moist heat, and I forgot to mention, moist heat is usually preferable because moist heat penetrates really well. Okay, you know, you could never stick your hand into, you know, boiling water, right? But you could stick it, you know, briefly into an oven that was super hot, you know, and it wouldn't hurt you. So with the aid of water, that heat is going to penetrate a lot more quickly. So usually that's preferable if whatever we're trying to disinfect can be around water. Okay, we've mentioned pasteurization a couple of times now. Of course, we talked about Louis Pasteur. Like I said, this is a sanitation that will kill microbes, kill pathogens, disease-causing microbes. We mostly use it to cut down pathogens in milk, but lots of other things like apple juice, ice cream, yogurts sometimes are pasteurized. The method that is most often used to pasteurize today is called flash pasteurization. Okay, so traditionally, when we first started pasteurizing milk, we did it this historical way. So you brought the temperature up to 63, again, well below boiling, but hot enough to kill all the disease-causing microbes. Thermophiles, you don't care about those. They're not going to cause infection for 30 minutes. Like I said, today, normally we employ this flash pasteurization. You bring the temperature up a little bit hotter, and then it only takes about 15 seconds. And typically it's cooled right back down again. So, you know, it passes through some kind of pipe, heated up, and then cooled down again. Overseas, it's a little bit more common doing these ultra high temperature pasteurizations, or what's called an ultra high temperature sterilization. You know, they're very similar, the temperature that's brought up there, but I believe by either one, or it may just be this sterilization, by that mechanism, you can keep milk out at room temperature. So if you've been to a grocery store in Europe, you might have seen all the milk out at room temperature. They can do that because they've basically sterilized the milk, and it's going to be good that way for months. With our flash pasteurization, we still have to refrigerate. Again, it's not gotten rid of all the microbes, just most of them. So it extends the shelf life quite a bit than if you didn't pasteurize it, but we still have to refrigerate it. So that seems like that would be a real advantage, not having to refrigerate the milk, you know, as we transport it to grocery stores and at home. But it hasn't caught on in this country because alters the flavor a little bit. So again, it just hasn't really caught on here to do it, you know, heating it up that high. Okay, I'm intrigued by pasteurization, so I'm going to spend a little bit more time just sort of FYI. So I have a couple of slides here that I didn't post, and you know, you're not responsible for this information, but I always like to think about the difference between raw milk and pasteurized milk, right? So raw milk is when it's straight from the cow. We often homogenize milk as well, and that means we just mix it up a lot. If raw milk was allowed to just sit, it separates out into like the cream and the whey and everything, and that turns people off mostly these days to drink it, so they shake it up real good. But if we were to just, you know, if you were presented with the option, here, would you drink this raw milk, or, you know, would you drink it, or would you say, no, it's raw, it's not been pasteurized, I'm not going to drink it? Is it safe to drink raw milk? So in thinking about that question, I usually ask students that question, and they, you know, click her in, and we say, oh, okay, would you drink it? All right, well, let's think about what we would need to know to make a good decision there. Well, milk is a really rich source of nutrients for microbes, so they grow really quickly in it. And there's lots of sources of pathogens. In a healthy cow, that milk is sterile, but then as soon as it comes out of the cow, there's all sorts of places where it can become contaminated. So, you know, the udder is dirty, right? You know, they're laying in their poop all the time, right? So just from the environment or milking machines, equipment, it can be cumbersome to clean out. So that's a source of contamination. You know, if the handler is hand milking it, dirty hands, whatever. So, you know, there's some overlap here, but there's lots of chances for that milk to become contaminated. Contaminated with what? Oh, there's a whole list of things, possibilities here. Number one is this Mycobacterium bovis. That's the kind of tuberculosis that cows get. If we drink milk that's contaminated with that, then humans can get tuberculosis. So before we started pasteurizing milk, it was a huge source of tuberculosis. Some other things that probably aren't too recognizable, anthrax. You get anthrax from drinking contaminated milk. Botulism, that's another bad one. Diphtheria, of course we vaccinate against that now. But before we pasteurize milk, you know, these things plus lots of others were common contaminants. Listeria, that's another one that can cause pregnant women to miscarry. So, so for these reasons, the CDC has called milk the world's most dangerous food source. So it sounds like I'm trying to convince you that you should never drink raw milk. But actually today, our cows are very healthy, at least in the United States. They're vaccinated, so they don't get sick. Farmers want to keep their cows healthy because they produce more milk when they're healthy. A cow will produce up to 8 gallons of milk a day. It seems like a ton. So cows are pretty healthy. They are milked in very sanitary conditions. So for that reason, actually, if you look on the CDC website, they say that it's actually okay to drink raw milk. As long as you're healthy, it's pretty safe. Now, if you're immunocompromised anyway, or if you're pregnant, they say you should not drink raw milk. But for most of us, it's fine. We started pasteurizing in this country around the turn of the last century. So by the 1930s, about half of the milk was pasteurized. Michigan was the first state to pass a law. In 1947, milk had to be pasteurized before it was sold. So a lot of your grandparents may have, you know, grown up drinking raw milk and they survived, but it's just sort of interesting to explore all that. Again, that was just a little extra information there for you on pasteurization. Questions, comments? Okay, the third method that I wanted to bring up of using moist heat, very commonly used, is an autoclave. So we use autoclaves all the time in the lab. We pretty much sterilize everything in the lab with an autoclave, glassware, our media, you know, pipettes, swabs, things like that. An autoclave uses steam under pressure. All right, so here's a picture of what an autoclave looks like. They're kind of old looking contraptions. They get kind of dirty with use, but you can see this has to be sealed around the outside here because you are increasing the pressure in there. When you increase the pressure, you're actually adding an additional atmosphere of pressure. Atmospheric pressure is 15 pounds per square inch. We add an additional atmosphere of pressure. And when you do that, you increase the boiling point. And we could have a discussion later about why that's true, but it has to do with keeping the molecules from moving around. So you increase the pressure. So we can increase the temperature of the water up to 121 degrees Celsius. Remember, normally it boils at 100. So typically that would be impossible. So we increase the pressure and we keep those parameters for about 15 minutes. It depends on what you're trying to sterilize. If it's like a lot of liquid or a really thick object, you might have to do it for a little bit longer. But generally, that's good enough to sterilize, you know, whatever it is that you're trying to keep the microbes down on. Okay, of course, you can't use it on everything. If there are materials that are not heat resistant, obviously you can't use it. Cutting instruments tend to kind of get dull over time when we put them in the autoclave, or if they're metal, they're going to corrode over time. Things like oils or powders are not good for the autoclave, but you know, when you can use it, this is a great method, again, to sterilize. And if you're interested in, you know, you don't need to know really how it works, but there's all these pressure gauges and, you know, steam coming and going, basically. Okay. How do you know if the autoclave is coming up to the correct parameters to sterilize your equipment? Typically, what we rely on in the lab is autoclave tape. So whatever you're going to autoclave, maybe it's some trash or something, you put the tape on, it looks like this. There's some kind of white lines on it. And then after you autoclave it, it comes up to the correct temperature and everything, those lines turn black. So you may have seen that around in the lab, the indicator in there. If you want to be absolutely sure, maybe you have a really important experiment coming up and you need to be absolutely sure your autoclave is working correctly, you might use a biological indicator. So we've used these in the past. Our autoclave breaks down regularly up in the lab. Basically, you have a plastic vial here, and inside it is a glass vial that contains some microbiological media and a strip of endospores kind of there between the glass vial and the plastic vial. So you autoclave that, and then after you autoclave it, you twist it so you break that plastic, you don't break the plastic vial, you break the glass vial inside of it so that the media comes in contact with that endospore strip. Then you incubate that for a day or so, and if those endospores have germinated, if they were viable, they will start to metabolize the media, and you will see some acidic end products as a result of that. And there'll be a pH indicator in there that will tell you that some metabolism is taking place. So if the indicator changes color, then you know that those endospores were not killed. You know that your autoclave is not working the way it should be. If it doesn't change color, then your autoclave basically rendered those endospores not viable anymore. Okay. All right, so we talked about moist heat. There are some applications of using dry heat. Again, for objects that maybe can't get wet, you might use a hot air oven. The drawback is that it requires hotter temperatures and it takes longer. Again, doesn't penetrate as well. But for things that you can't get wet, that might be a good option. So usually the parameters, 160 degrees Celsius, that's only about, I think, like 320 degrees Fahrenheit, something like that. So, you know, that's your oven at home. Certainly, you know, you put something in there for a couple hours and it should be sterilized. Of course, some things are gonna maybe perhaps catch on fire, like paper, you know, some, you know, rubber things you can't, you know, get hot for that long. So again, there's some things that it's not gonna be useful for. Another dry heat application is just burning something. Obviously, we do that with trash a lot. You do that in the lab every time you flame your loop. All right, a lot of things we can't burn and have them still good for what we want to use them for, but flaming your loop, again, is just one example. All right, what about using cold to cut down on microbial growth? Refrigeration and freezing, like I mentioned before, this is a, if we're talking about, I usually focus on bacteria. It's bacteriostatic, okay, meaning that it's just going to slow or reduce bacterial growth. Okay, that's why we refrigerate our food, okay? There are some exceptions to this, of course. There are organisms, psychrophiles, that grow best in cold temperatures. We usually define them as growing best below room temperature. Okay, your refrigerator is, I think, around 40 degrees, about 4 degrees Celsius. There are some organisms that grow really well in your refrigerator. One example of that, I mentioned it briefly as a sometimes contaminant of milk, is Listeria monocytogenes. And this organism is not actually a psychrophile. We actually call it a facultative psychrophile because it actually grows faster at room temperature, but it grows pretty well at refrigeration temperature as well. So this is a fairly common cause of foodborne illness. When you see outbreaks, oftentimes it's because of Listeria. So if the food was contaminated, you know, at the processing facility, and then, you know, consumers buy it, throw it in their fridge, it's growing up that whole time. And then when you ingest it, typically for somebody that's healthy, they might get a little bit ill from that. But like I said, it can cause miscarriages in people that are pregnant. So sometimes doctors would tell pregnant women to stay away from any foods that might commonly be contaminated with Listeria, like cheeses that aren't pasteurized or maybe lunch meat. There was a bad outbreak of this in the Bill Maher packaging facility in Zealand in the late 90s. About 20 people were killed. So I think there was an outbreak in cantaloupes about 10 years ago that killed about 20 or 30 people nationwide as well. So, okay, freezing, again, this doesn't kill microbes. We actually freeze cells if we want to store them long term. We'll freeze them even at minus 80 degrees Celsius. When we thaw them out, enough of the cells are viable that, you know, we can start new cultures there. So, okay. All right, and then other methods that I'll just mention fairly quickly. Oftentimes, well, or I should say, bacteria need water in order to metabolize. So if we deprive them of that water, then that's going to kill them. So, you know, just desiccating, drying, say, food out. Here's a bunch of apricots drying in the sun. That's going to keep the bacteria or the fungi from rotting the food. Another method, basically kind of the same principle of depriving the microbes of water, is lyophilization. So another name for that is freeze drying. You might have eaten freeze-dried food. If you were a backpacker, maybe here's some astronaut ice cream, or basically to eat it, then you just add a little water, and apparently it's pretty tasty. But with this process, cells are frozen pretty quickly, like maybe in liquid nitrogen, something like that. And then that frozen water is removed by a vacuum. So this is a process of sublimation. The water is going directly from a solid to a gas, skipping that liquid phase. All right, so if we want to store cells or food, we can do that indefinitely, again, because the microbes don't have water. All right, filtration is another method. If you're trying to sterilize something that can't be heated, so maybe something like a vaccine or some kind of drug like an antibiotic, maybe some kind of enzyme or something, you can put that solution in a filter vacuum. There's a membrane right here, a membrane filter. You can have pores so small that they won't even let viruses through. Of course, it's not going to let the water through either, so you have to suck it through that filter by vacuum, and then that's able to sterilize that medium. About 10 years ago, you might remember hearing about an outbreak of fungal meningitis. And they traced it back to a pharmacy in New England that hadn't sterilized. It was a steroid drug, prednisone, that people actually got by injection into their spinal fluid. They were having back pain. Well, that drug hadn't been sterilized properly, hadn't been tested, and people were injecting fungi, I think it was aspergillus, getting that injected right into their spinal column. So a number of people died from that, just failure to sterilize that drug properly. And then we talked about osmotic pressure, as well as another way that cells are dehydrated. So we add lots of some solute, usually salt or sugar, to food in order to preserve it. That creates a hypertonic solution that those microbes are in. They dehydrates them and they don't spoil the food. So usually something like jam or jelly, it's eventually, right, they're going to spoil. It's usually not by bacteria. Bacteria like salmonella are going to really not withstand that hypertonic environment very well at all. Fungi are a little bit more able to survive in that kind of environment. So usually it's molds that are going to grow on your food a lot quicker than bacteria. All right, so the last big physical method that I want to talk about is the use of radiation. Okay, and we have two kinds of radiation. We're going to start with ionizing radiation, and then we'll talk about non-ionizing radiation. So what is that exactly? Little reminder here of what visible light is. So the wavelengths of visible light are between 400 and 700 nanometers. I think it was just last week, I saw a rainbow all the way across the sky. So the red light, little longer wavelength, that's always on the top of the rainbow. The purples and the blues are always on the bottom part of the rainbow due to its wavelength. Okay, so above visible light, we've got our infrared and our radio waves, our microwaves. Shorter than visible light. Okay, so what I mean by wavelength is the distance from the trough to the trough or the peak to the peak. Okay, that's the wavelength. And as a wavelength gets shorter, it's going to increase, increasing energy. that kind of cut out there a little bit on mine. But as wavelengths get shorter and shorter, they carry with them more and more energy. So we can use ultraviolet light, that's wavelength somewhere between 1 or 10 and about 300. We'll talk about that in a minute, in order to kill cells. And shorter than that, X-rays and gamma rays. Okay, so we're going to talk about the effects that those have on cells. Electron beams can also basically fire electrons that would cause ionizing radiation. Okay, so we call wavelengths this short, X-ray, gamma, and gamma rays, we call them ionizing radiation because the effect of that they have on cells actually create ions in cells. And so basically, those wavelengths can actually break DNA. If one strand of DNA is broken, usually a cell can fix it because it has a template to work from. But when it breaks both strands, it's really hard for the cell to fix that because it doesn't have a blueprint to do that accurately. Those wavelengths can oxidize covalent bonds. So there's lots of covalent bonds in proteins, nucleic acids as well. An ion is going to be stealing electrons from other molecules, so causing all sorts of damage there. Even creating hydroxyl radicals. So that's an OH with an unpaired electron. You might recognize that. That is the most toxic of our toxic oxygen forms. So again, it's stealing electrons from lots of other molecules. So ionizing radiation is causing all kinds of cellular damage to DNA and proteins and all sorts of other things. Okay, it doesn't heat up an object, so that's good for objects that can't be heated. We can sterilize them with some kind of ionizing radiation, and it kills cells really quickly, much more quickly than if we use non-ionizing radiation. And it penetrates really well, so it can even penetrate through solids, right? X-rays, you know that. All right, so if we look at some applications, ignore the Hulk there for just a second. So some forms of ionizing radiation and what we might use that for. All right, so I mentioned using an electron beam. Okay, where does that come from? Well, we have special machines called cathode ray machines that can produce electron beams. It kills cells really quickly, so that's an advantage. But unfortunately, it doesn't penetrate very well. So it's mostly for surface sterilization, and there's some applications there, some medical supplies like bandages, or even some spices, or some different kinds of food. Probably what has an even greater possibility of being in much more widespread use are gamma rays. Okay, where do gamma rays come from? They're emitted from some kind of radioactive element. I think cobalt-60 is a common one. So again, the gamma rays do come from radioactive elements. What could we use them for? They have a lot of potential to be used to keep our food supply safe and extend the shelf life. So here's a nice picture in your book. Both sets of these strawberries are about 2 years old, or sorry, 2 weeks old, I believe it said in your book. This group here on the right was hit with some gamma rays, and this group on the left was not. So that's how they look 2 weeks later. So you can see that the group on the right isn't spoiling at all, and the one on the left is rotting away. So we have real potential to increase the shelf life. I can't even imagine how much food grocery stores have to throw away, that it just sits there and rots. Foods that have been irradiated, you're supposed to see this symbol on them, this little flower symbol. And there's only certain foods in the US that have been okayed to be irradiated with gamma irradiation, because there's a little bit of pushback from the public. This is kind of slow to be accepted because people think that it makes their food radioactive. All right, so this is not, you know, like Bruce Banner struck with a blast of gamma irradiation and then he turns into the Hulk every time he gets mad. That's totally not correct, right? It doesn't make the food radioactive. People that don't understand science just kind of believe whatever they want. It doesn't leave any residue at all. It can change the flavor of the food slightly. So it kind of depends on what kind of food it is. It changes the flavor maybe a little bit, and it does decrease a couple of the vitamin contents just a little bit, but pretty nice trade-off that it doesn't spoil as quickly. So like I said, in this country, only certain foods can be irradiated. In other countries, basically anything goes. They have laxer standards about that. But if you think about a vegetable like spinach, it's, you know, something that's sold fresh, right? You can't heat it to cut down on any foodborne pathogens. It's just a great tool that we could have to, you know, hit it with some irradiation. If the spinach field has been contaminated with some water, you know, that's been contaminated by some livestock runoff, we've got E. coli on the spinach and it's sticking to the spinach with its fimbriae. It's hard to wash off. So again, if we could hit it with some gamma rays, then we would, you know, ensure our food that it's a little bit safer. Penetrates better than what the electron beam does. And so again, use to protect our food supply is one use. Other medical applications like implants and things like that, it's a good way. Better than using chemicals and some things that you can't heat. Okay, there's not as many applications to using X-rays. It penetrates really well, but it takes a long time to kill microorganisms. So there's not as much use there. Of course, we have to be careful because gamma rays and X-rays and electron beams are going to be harmful to us. So we have to be careful, like, not to expose ourselves to any of these things. And of course, it's more expensive to use than what you would in your home, of course. Okay, so contrast that to ultraviolet light, which is considered non-ionizing radiation. Okay, so remember, this is from about 1 nanometer to about 300 nanometers. It's considered the ultraviolet range. And ultraviolet light causes a very specific kind of mutation, and that's why it's harmful to cells. It causes thymine dimers to form in the DNA. Okay. And it's most often thymines just because of the structure of that base. Can happen in cytosines too, but it's usually some pyrimidine that causes that they bond to each other when they're exposed to UV light. So here we have some DNA that's getting hit by UV light. This is one of the reasons we spent some time on the structure of DNA. Okay, typically you would never have two bases on the same strand bond to each other. They're supposed to be bonded to their complementary base on the other strand of DNA. When you have a dimer, so two bonded right next to each other, you can see how it causes this out-pocketing of the DNA. DNA polymerase 3 that was going through to replicate that strand of DNA probably is not going to read that correctly. There might be some insertion or deletion there. If this strand was being transcribed, RNA polymerase probably wouldn't read that correctly. So all cells have repair mechanisms specifically to fix thymine dimers, enzymes that go through and fix those. But eventually those repair mechanisms can't keep up. So if you have a long enough exposure to UV light, that's eventually going to kill the cell. And of course, it's harmful to us as well, mostly in the form of damaging our skin cancer, sunburn, that sort of thing as well. Okay, so the one drawback to using UV light in order to, as an antimicrobial, is that it doesn't penetrate very well. Penetrates really well through the air, not so good through liquids, really not at all through solids. So it doesn't penetrate as well as ionizing radiation. And again, we do have to be careful. There's a few more applications of using UV light though. You can buy like toothbrush holders that will hit your toothbrush with UV light. I think in the lab here, sometimes we sterilize chem goggles with UV light. Lots of applications to treating liquids. So I didn't post this picture, but just to show you, most sewage treatment plants use ultraviolet light as a last stage of cleansing before they release that water, say, out into the Grand River. So they do this in Grand Rapids. I don't think we do this in Allendale, but I haven't visited the sewage treatment plant in a number of years. But I know about 10 years ago, they switched in Grand Rapids. They have to regulate the flow of the water really carefully to give that UV. These are all the UV lamps right down in the water there, give it enough time to kind of penetrate through the water to kind of reach all the water. But again, doing this instead of dumping a bunch of chemicals in the water seems like a pretty good option. Another application of UV light, and I haven't talked to anybody that has seen this in Grand Rapids hospitals. This was just a picture I got off the web. I think it was in a Denver hospital. Just one more tool to sanitize patients' rooms. So you wheel this UV light into a room. You can see how they've opened the drawers here just to try to get that light to hit like every surface that they can. They leave the room, turn it on remotely, leave it there for 15 minutes, turn it off, come into the room, move it somewhere else in the room to just hit every surface you can. Especially maybe a patient that had an antibiotic resistant infection or C. diff. That's a diarrheal infection where the spores are excreted. So it's a little bit harder to kill. So one more tool. You can't get chemicals in every nook and cranny. So again, it's just another tool that we have. Okay. All right, so that's our physical means of microbial control. Okay, so we're going to start on our chemical ones. I know this information is a little bit, to me, it's a little bit drier. Hopefully it's not too hard to understand anyway. But in terms of kind of thinking again, some of the things that we thought of before about antimicrobials in general, but if you're thinking about a good chemical to use, there's lots of things that you want to think about. Does the chemical have any side effects? Okay, we want a chemical that's toxic to the microbes and not toxic to us, for sure. That's a big consideration. Is it soluble in water? And is it useful at high dilutions? Okay, both of those are going to determine how expensive it is. All right, if it's going to be used widespread, it's better to have something inexpensive. What kind of organisms does it work on? We call that being broad spectrum if it works on a wide variety of microbes. Okay, obviously that's ideal. Okay, and again, emphasize, ideally it's going to be effective in a high organic load. Okay, so ideally we could just spray it on, you know, whatever the mess is that we have to clean up, and then, you know, kind of clean it up all at once. Okay, so none of these chemicals are perfect. I want you to think about, in particular, how they work, the uses that they have, and if there's any drawbacks to using the chemical. So I'm going to give you a little bit more information about that than these chemicals, but those are sort of the main things that I want you to keep in mind. I tell you some other things about them just to try to make the conversation a little bit more interesting. Okay, and I have the chemicals arranged, like I mentioned before, from low level to high. The high level ones are the only ones that are going to kill spores. Again, the lower level ones are working against the kinds of microbes that are a little bit easier to kill. Okay, so we'll start out with soaps and detergents. Probably the most common thing that you would come in contact with. Okay, what's the difference between a soap and a detergent? They kind of seem like the same thing. A soap is supposed to be derived from natural products, and a detergent is synthesized. So that's the difference. They both work the same way. They are surfactants, and that means that they reduce the surface tension of liquids. So they're really good at kind of breaking up dirt and washing it away. And their main thing that they're going to target is disrupting cell membranes and cell walls. Okay, so an example of a detergent is quaternary ammonia. So you probably have heard of them. Most of the time we just call them quats, commonly used in hospitals and lots of other settings. This is an example of the structure of one, just in case you're interested. Unfortunately, like I said, it's low level, so it's not going to work on endospores or naked viruses even, or mycobacteria, but they are pretty effective and they don't leave any residue. So they're widespread, have widespread use. The one drawback is that we are starting to see some resistance to especially quaternary ammonia among an organism called Pseudomonas. And Pseudomonas is a pretty common hospital-acquired infection. So quats used a lot in hospitals. We're starting to see some Pseudomonas that are, it's a gram-negative bacteria that are not sensitive to quats anymore. I've also found Pseudomonas, like I had my lab go around and swab different places on the 3rd floor of Henry, and then we streaked it on some nutrient agar and see what grew up. There have been cases reported of Pseudomonas being able to be cultured from soap scum. I tried that and I didn't get anything to grow, but so we are seeing some resistance, and that's one drawback. I will say that it's kind of disheartening today. It's really hard to find a soap that doesn't have some antibacterial chemical in it. In general, our society is kind of crazy about killing every little microbe we can, and it's kind of understandable now in a pandemic, but even before that, we get a little bit crazy. We can go too far in trying to kill everything, and then we're breeding things that are resistant. So recently we have seen some chemicals being outlawed, again, because they're just not really necessary. So that's something that we'll, I guess, continue to wrestle with. Okay, I think that's a good place to stop. We'll pick up next time talking about the heavy metals and probably get started on our antibiotics. So I'll be posting something about that this weekend. Have a good weekend.